Hello, everyone. So for this video, I'm going to show you how to do the blood typing simulation lab, and then I'll give you the results of the case that I'm running for you. So the first thing you're going to have is the worksheet, and on this worksheet, you can see that you have your chart to fill in as you move through this. So those that are virtual or at home, that's what you'll be filling in as I go through this, through the results. Okay, so what we look at here, I'm just going to take the camera and kind of pan over. What we have here is your blood samples. So we have an unknown 712B5. We're going to have 4 suspects. We're going to have Denise. We're going to have Bonnie, Tara, and Candace. Okay. The other things you're going to have is a well plate, and on this well plate you have an A, you have a B, and you have an RH. And with that, they're kind of nicely labeled. A little tough for you guys to see here. And then in order to test your blood, you're going to have an A serum. It's called anti-A serum, which will test for A blood. You have an anti-B serum, which will test for B blood. And you have an anti-D serum, which will test for the positive or negative portion of your blood. And what happens is if when we put these in contact with the blood, if they do what's called agglutinate, which means they get little spots in them or they start to gel together, that means that that antigen is present. So if the A well, which would be right here, and I'll run you through the scenario. If the A well was to agglutinate, that would tell you that you have A blood. If it agglutinated in the B, that would tell you you have B blood. If it agglutinates in both, that means you have AB. And if it doesn't agglutinate in it, you have O blood. And then the RH factor, which we also call the D antigen, that works the same way. If it agglutinates in there, it means that it's a positive. If it doesn't agglutinate, it means you're negative. And I will run you through our unknown, and then I will show you the results of your 4 suspects. So what we're going to do, I'll see if I can pan you down and show you a little something here. Okay, so what we're going to do is we're going to take our unknown, and our unknown is unknown 712B5, and you put a drop in each well. In the A well, we're going to put our anti-A, and that's going to test for the A antigen. In the B, we're going to put the anti-B. Notice how we're keeping them separate right now. We don't want to mix all of the different testers because then we wouldn't know which one is causing a reaction. And then what we're going to do is we're going to take one of our toothpicks, and you mix the A well for about, I don't know, about 10 seconds or until you really start to see a reaction. And what we want to make sure of is we want to change out our toothpick for each sample. Here's the B well, because we don't want to cross-contaminate and have the wrong results come out. And then we'll do our D antigen. Okay, so all those have been sitting for about 30 seconds in total. And now I'll try to give you a close-up as best as I can. So the first well up here, let's see if we can focus this as best as possible. So this first well up here, you may be able to see that there are some dots. Glare is not helping me out that much. There are some little dots that are forming in there. So that would tell us that the A well is agglutinating. The B, which is down here, this one also has some dots in it. Oh, there we go. Now we can see it. Okay. You can see that there's some little red dots in there. So that would tell us that it's agglutinating as well. And then the RH factor, which is over here, this one is showing nothing. Okay, that one's very clear. So what that tells us is that we have no agglutination here. So if we look up here, we have spots that can be seen, an agglutination in A, we have agglutination in B, and we have no agglutination in the RH factor. So that would tell us that this blood type is AB negative. And that's what should be filled out in your chart. You should be showing that there's agglutination in the A, there's agglutination in the B, and there's no agglutination in the RH. Okay, so hopefully that was clear for you. So then what I did is I ran this already for you with our other samples. Okay, so this is Denise. And Denise is showing Denise is showing agglutination in A, showing no agglutination in B, and it's showing agglutination in the RH factor. You can see that kind of like clumping up. Okay, so Denise showed agglutination in A, no agglutination in B, shows no clumping, and agglutination in the RH factor. Our next one is Bonnie. We did the same thing. I put a drop of her blood in each of the 3 wells. And then we added our anti-A, anti-B, and the RH. So for A, we can see that we have agglutination. There's little spots in there. Okay. There's agglutination in B. There's little spots. Those are pretty tough to see, but they're there. And there's nothing in the RH factor. Okay, it's very clear. There is no agglutination there. So we have dots. We have agglutination in A, agglutination in B, and nothing in the RH factor. The fourth or the third suspect, our 4th sample in total, her name is Tara. We have no agglutination in A. It's very clear. You can see that B has agglutinated with these bumps. And then our RH factor is clear. There are no bumps in there. It looks like it's starting to just dry out a little bit if you're seeing those little air bubbles. Okay, but that's clear. So you only have agglutination in the B for Tara. And then for Candace, our last one, we have no agglutination in A. Very clear in there. There's no bumps, no bubbles. B, same thing. There's no agglutination. And in the RH factor, we have it turning into a gel. You can see it kind of clumping up. So that would mean that we have agglutination in the RH factor. Okay, so we have no agglutination in A or B for Candace, and the RH factor, we have agglutination. Okay, so you're going to use that data to fill out, or you should have been filling out your chart as I was walking you through that. And then you're going to try to decide on whose unknown blood was on the scene, and then answer the questions and try to fill in those, what I just went through with your plates. You're going to fill in those on your last page. Okay.